and position 140 is cysteine. And so formidehydrogenase messenger RNA has here the UGA codon, and the tRNA sec has the bound to cell B, the UGA, the anticodon that recognizes UGA. So what we really wanted to do to see how many nucleotides in E. coli can we recode by making 64 different codons here. So this is a typical synthetic biology experiment where after creating the right strain, we had to synthesize 64 messenger RNAs with 64 different codons in this position. Then we needed 64 tRNA cysteine variants with 64 different anticodon sequences in this position. And then the secret, of course, you have to find out, and we have always been very fortunate in this, that we get a very easy genetic test that is exceedingly sensitive. And a readout for formidehydrogenase is benzylvalogen. It's an old test that gives a purple color. And if you look, there are 60 out of 64 codons which you can recode. But then you really have to do it by a chemical experiment, learning from Govind. So you have to purify each FDH from each experiment, determine the enzymatic activity because only sec-containing enzyme is active, and then determine the sec content by ICP-MS and identify the position with LC-MS-MS. So with all of this, then we showed that 60 out of 64 codons can be recoded cysteine, some of them completely, and given the fact that they are now, if you then have these sense codons to do this, they are no longer hindered by any release factor. You can get protein increases up to 12 or 15-fold even. And we also found out that some sense codons may even be recognized by RF2, and this is a different analysis that needs to be done. Then I would like to give you a different kind of recoding. And this is absolutely opposite to everything that you have seen in literature by the genome engineers. And this goes via the supplementation-based incorporation methods. So if you have an isostructural non-canonical amino acid that tricks the amino acid tRNA synthetase to be charged, you then get protein variants where you get the analog instead of the wild type. So it's a natural substrate tolerance of the endogenous host synthetase allows this. And in this particular case, the aim was to replace tryptophan with cyanopyroalanine in protein synthesis. So the 6-membered ring that you see here of tryptophan would actually be replaced by the 5-membered ring with sulfur in it. And to make the long story short, this was simply done by a serial transfer experiment in batch culture, which after 280 transfers and doing some miracles with what you give the strain to grow on, you got a proteome-wide exchange of tryptophan for this cyanopyroalanine E. coli. The cell itself grows only 10% slower than wild-type E. coli. And so basically this has happened. And in essence, and I know the synthetic George Church and others are upset at the nomenclature, we have recoded 20,899 UGG tryptophan codons in this experiment by simply restricting the amount of tryptophanol tRNA tryp, or what that the cell has that can be used. And we are continuing these experiments with analogs that are far more distant from tryptophan to see what the plasticity of the proteome might look like. And I will give you the last experiment, and this was the question, how is a genetic code expressed in nature. This really has always been an interest, because when we in Corrales laboratory deciphered what the code is, many observations were made and many ideas were embedded in this that really were dogma and that couldn't be challenged. And the question was, is this really so? So can a vast amount of genomic and metagenomic sequence data provide information? We could, and we looked at over 10 terabytes of sequence, and the tRNA sequence structure is the key. And this was work done by Takahita Mukai, together with people from the Joint Genome Institute in Walnut Creek, California. And we took tRNA-seq as first attempt with the covariance model because it's a long and an unusual tRNA. And then one was just trying to sequence. So the major point is in here. This determines the anti-codon diversity of tRNA-seq. We got a vast number of sequences. There are many exciting examples that have come out that are different from what I'm going to show you here. But this is what we got for real selenocysteine. So we saw that these are codons that are used by various organisms for selenocysteine. And the take-home message is that selenocysteine in microorganisms, even eukaryotic microorganisms, is encoded by 10 sense codons and 3 nonsense codons. So the idea that you have a firm assignment is out the window. And one reason is, why is there selenium? And you can ask me later. One can speculate for a long time. If you have a protein that has a selenol moiety in it, it's oxidized very easily. If you have a protein that has a SH moiety, cysteine in it, it's also oxidized. The big difference is, with any sulfur reagent, you can reduce the oxidized selenoprotein, but you cannot reduce the oxidized cysteine-containing protein. So if an organism experiences an oxidative habitat, it may actually change and encode for cis-selenocysteine because it will help the organism to survive. This is just one example. So with this then, I would like to leave you with that the likelihood of discovering a new amino acid present in all 3 domains of life is exceedingly low. I don't think it's going to happen because we analyze sequence so many already. But a new amino acid localized to a small group of organisms is possible, and this is of interest, like with pyrrolysine. So if you need a certain pyrrolysine evolved because a certain enzyme that takes up trimethylamine that is good for the energy metabolism of the cell, that enzyme is pyrrolysine in the active site. The exciting thing is, when you think about it, once this happened, the organism could use a pyrrolysine for anything. So we find now organisms that are no longer requiring pyrrolysine for a particular enzyme, but they just use it, and this is really how an amino acid comes into existence, so to speak. If a new translational scheme evolved, nature will use it anywhere when needed. 15 or 20 years ago, we made big discoveries that we said are only in archaea, and everybody said it's absolutely correct. Archaeal protein synthesis is very different from eukaryotic protein synthesis. But then turn around and look at it now, it's not. So there has to be a need for deeper understanding of cellular metabolism, because the relationship of codon usage and organismal fitness is something that will determine how many of these engineered organisms can be made. code evolution is possible. The fitness of organisms containing additional amino acids will be decreased. But the take-home message, the genetic code is far more flexible than we previously thought. And if anyone of young people here give my lecture in 15 years' time, it will be different again. So I have to thank all my colleagues. These are the people in my lab: Anna Čanković from Croatia, Felix from China, Kyle from Canada, Huisi Kok from Singapore, Sergei from Russia, Takahito from Japan, Noah Reynolds from the US, Anastasia from Russia, Tateki Suzuki from Japan, and Oscar Vargas from Venezuela. I was fortunate to collaborate with Nedilko Bodisa from Berlin, David Bryson and George Church from Harvard, David Liu from Harvard, and Nikhil Peters from the Joint Genome Institute, and my former postdoc, Eric Wang, who is at the Academia Sinica in Taipei. And I acknowledge happily support from Department of Energy and National Institute of General Medical Sciences. Thank you.
Okay, thank you, Dieter, for a really enlightening seminar. I think we have time for a couple questions.
Dieter, with a highly evolved organism that uses this tryptophan analog, have you done the genome sequence or something like that to see what's the big change, or what is responsible for this now?
So the question is really, do we know anything why this organism is still happy? To our amazement, there are not more than 150 mutations in it. There are 2 mutations in TRPRS to make the TRPRS better. But we also have got some insight that hopefully we'll have in the future, that there are certain gene changes which will make this evolution 10 to 15 times faster.
So if you add
in this case, it is not toxic. For old people have memories. In 1983, there was a paper in PNAS to show that certain bacteria, Bacillus bacterium, had fluorotryptophan in the DNA. And this leads to Tom's question. In that case, it was shown that tryptophan was detrimental for the growth of these organisms, because only fluorotryptophan was accepted. And I will tell you then one other story, and I forget for the audience, I forget the name of the person. If then a synthetic biologist comes along and does an experiment, he or she should be cleverer than nature. And this is what we learned from Gobind, what the synthetic biologists did not learn from their advisor. Because Gobind said, whenever you get a chemical, you analyze and look what else is in it. And so there was a paper published in 2005 to regenerate this fluorotryptophan evolution. And the people did glorious work and even put in a paper that the fluorotryptophan had 80 micromolars tryptophan in it. And all the results, of course, were just described, gave wild-type E. coli. Wild-type E. coli, because E. coli knows how to distinguish tryptophan from fluorotryptophan. So this is something that I think in modern times we don't teach students anymore. Gobind insisted that you are responsible for the compound that you use. And I don't want to go into examples. This is absolutely essential. Today you buy a kit, you assume that the kit is fine, and that's it. Sorry. Marshall?
Thank you very much. I have small two queries. With 3.8 billion year history, with all permutations and combinations available under different sets of conditions, which have changed tremendously in every eon, nothing out of this possible combination has been left as a fossil in any of the organisms. They have not been able to carry that one. And do you expect that in the remotest environmental harsh condition or something, you are likely perceive that you will encounter such a thing somewhere? And second part is that two new codons, do they connect with this part in the synthetic biology story?
These are very good questions. I mean, what I, as a simple chemist, didn't realize, that in evolution, you come to a fork, and this organism just grows infinitesimally better than that organism, but then evolution goes this way. So you only have a very small difference to change. Yes.
It does not have a direction.
So
it goes towards simplicity.
This is correct.
This is after 100 years, Homo sapiens won't be able to synthesize glycine.
Yes.
Yeah. And the codon of this one will become redundant. Okay.
So
where
you mentioned
it after
the environment is very important.
Yeah. Agree.
And the environment will drive it.
Yeah. But nothing combination of this one is available in any relic form, any fossilized form, or any of the, in this solar system, which is mine.
I think you or I, we are very likely old enough to look for things like this. They're considered crazy ideas by people. And I will give you one personal. I go to the gym with Tom Stites, a Nobel Prize winner, and I asked Tom, I said, Tom, I think glutamine is the last amino acid. Can you imagine what life would like without glutamine?
Without?
Glutamine.
Okay.
And he said, this is an idiotic question. Don't you know how important glutamine is in protein structure? So I said, yes, today, but heaven knows.
Not tomorrow.
Yeah. So I think your questions should be asked.
Okay, any other questions? Yeah.
I have a few queries. The first one is your demonstration of incorporation of tryptophan analog at a specific position is not that amazing, because we know in proteins, tryptophan from the structural point of view, it has very little functional role as such. So mostly it's in the core.
This is true. This is why we use tryptophan.
Yeah.
On the other hand, and this is what my NIH program director calls it, but these look, there are so many papers show that these three E. coli proteins have essential tryptophans in it. So we are actually doing the structure of these proteins, but it will be very likely, as you said.
Because it's most likely the distribution is in the core and the packing.
Yeah.
Right? Because in protein crystallographers often do use auxotopic strains and incorporate selenomethionine for phasing experiments. So we can take a auxotropic strain of E. coli, which can't manufacture methionine, and put selenomethionine. All the methionine distribution is quite wide on the surface, and so which can be problematic. Sometimes it's a problem. Another question is related to your N-terminal domain. I'm very curious, you wrote in the slide insoluble. Is it insoluble, or you want to infer unstructured?
When pyrolysis, let me go back one. Pyrolysyl synthetase is an archaeal enzyme. So the organism it comes from has 1.1 molar sodium chloride as a normal concentration in it. In high salt we can purify it easily. If I go to below 0.8 molar, it just precipitates. So when I said insoluble, it is in our laboratory conditions that everybody uses.
So you give that, you can express the domain independently, N-terminal domain, and put in those conditions, and you can look at least the solution structure of it.
We did it and we got the structure. We got the structure and it helped that we actually put zinc in it, because we had no idea that it was a zinc protein. Thank you very much for your comments.
Okay, I think it's time to move on, especially since we've now established that as you grow older, you get crazier, but you can still do great science.
Can I ask one question from sir?
Okay.
Ma'am, thank you, sir. So my query is that you told that the SH group, and that is because of cysteine in proteins. So is that SH groups can affect the expression of the protein or their solubility? If a protein having a high SH group, higher number of SH group, that is a cysteine, does it affect its expression level, or I should say, secondly, its solubility?
We do not know. And today, of course, you also have the availability of other expression systems than E. coli. So I'm sure if you have a very difficult problem you would like to express it, you can find systems where you can express it. For us, it was simply established methods, E. coli, and we have not looked.
Okay, thank you.
Okay, let's thank Dieter once again. All right, so our next speaker is Umesh Varshney, and he's from the Indian Institute of Science in Bengaluru, and his title is Pioneering Round of Initiation Complex Formation Triggers Ultimate Steps of rRNA Maturation in E. coli.
Good morning, everyone. It's really wonderful to be here. Meeting a lot of people here from the old time. I'd like to thank Tom and Rajendra to put it together, and the Team India to really do a marvelous job that we are all enjoying here. I joined Khurana Tom group towards the end of '88, December '88, and I was there until July '91. And here is an undated picture that I fished out from my computer last night. I think it must be 1990, because I see here Nripen Mandal. I think he joined a little after I did, and so I think the picture must be 1990, and you can see Sriram here. I can't see from here. I think somewhere here it must be there with his beard on. We listened to him yesterday. Of course, Govind and Tom. Julia is over there. Anne-Marie is also there. So Anne-Marie was really the person taking care of everything in the lab. There is another picture I have. This is in the new building, and that must be in '94 actually, because I was visiting. 95, no, we moved in '94, I think. That's the time I was visiting, and I happened to be there around the time that, group pictures are taken, so I'm right there. So again, I think, Ramesh was there at that time, so I guess, it must be around that time. There are a lot of things, I've learned from both Tom and Govind, and really, they've really shaped my life. Before I came to MIT, I knew how to do science, didn't know why I was doing it. But, coming to the group was really wonderful because, one could start asking questions, that would really mean something people would find them interesting. And so I think, thank you very much, Tom. We don't thank you enough. So it's really, it was lucky for me to be there. And, small encounters with Govind were also very useful. He made me realize what science is. One day I was in the kitchen making my coffee. Govind walks in. He asked me, what I was doing. I said, I'm making mutants. Well, that's our culture. So really, I think the way he was looking at things was really quite, all into science all the time, and that's really changed things. So I think in that year, I also sort of, this is a picture in a restaurant actually, somewhere close to Tom's home, and I think, so it was really nice to visit at that time. And then a few years later, we had a symposium for Tom that was in 2011, and many of us were there at that time. And so thank you once again, Tom, to let us have that. In fact, I think Asim yesterday made a proposal. I'd like to second that in public. So with that, let me now begin the talk. And what I'm going to do really is some of the slides that you're going to see are going to be the slides that I've used in my Monday seminars and also in Thursday seminars. Govind used to have Friday lab group meeting, and Tom was having them on Thursdays. So a lot of these slides are going to be common. And this particular one is really one of the very favorite ones, and I'm not sure what Tom is going to have in his talk also. But this slide really tells that in the cell, there are two kinds of tRNAs. One is really the garden variety, I would call it, elongated tRNA, which is amino acylated. Then with the help of elongation factor TU, it goes to the ribosomal A site. Whereas the other one, which is a special tRNA, the initiator tRNA, is amino acylated with methionine tRNA synthetase, and the formulated tRNA then goes to the ribosomal P site. Now this is the only tRNA, in fact, that binds directly to the ribosomal P site. All other tRNAs must first bind to the A site here and then moved on to the P site. So what that really means is that there are a few questions that can be right away asked from this observation. And that is, how is initiator tRNA selected onto the ribosome? And secondly, how does it participate in initiation process? Now, this is the initiation pathway that has been known for some time. And what is really happening is that much of the important activity in initiation is happening on 30S ribosome. So you have all the initiation factors bound there. Initiator tRNA is there. 50S subunit joins. And then something happens. All of the initiation factors go away. Initiator tRNA occupies the right place. And we have elongation competent 70S ribosome. Now much of the things that are here in this black box are not really that well understood. And the point here is that I'm trying to make is that through all of these stages, initiator tRNA is right there. So one could in fact even use initiator tRNA to understand each one of these steps. So this was the idea that I had when I came back. And of course, we are using the initiator tRNA. So let me tell you something about the initiator tRNA. In E. coli, there are 4 initiator tRNA genes. 3 of them are at 63.5 minutes, and they're part of a single operon. The 4th one is a little away, 71.5 minutes, and there's a single tRNA gene. All of these tRNA genes, in case of E. coli B, in fact, are identical, but K, there is a single change here. But without worrying about that change, the overall structure of the tRNA is shown here. And there are 2 parts that Tom had really established to be very, very crucial for this tRNA. One,